be maintained. Okay, and that's the other thing I was just going to say. Recording has been initiated. So these sessions or these lectures will be recorded for all. If you have a question of one of the speakers, please type your question into the Q&A space. We've activated the voting system. You can check the Q&A and vote for your questions that you want answered. The second piece is if you have a technical question, you can use the chat function to contact the organizers. But we will not monitor this chat space for the questions to the speaker. I already mentioned that the sessions are going to be recorded, and we will go ahead and get started in the interest of time. So our first speaker comes to us from Iowa State, Dr. Chris Tuggle, and he's going to be talking about his experiences, pros and cons, I would say, of cell type expression phenotyping from deconvolution of bulk RNA-seq data. We're all aware that this is a key aspect of trying to connect genotype to phenotype, particularly for SNPs associated with controlling gene expression. And then obviously we would like to take that down to single cell resolution. So Chris is going to talk a little bit today about his experiences in the pig. All right, Chris, the floor is yours.
Okay, thanks, Wes. One thing that we could add is Sunita's going to put a link into the chat later. That is a very short survey with a couple of questions that we hope that you answer before you leave the call. And that will help us understand how, whether we should add more of these field days or how we can improve that in the future. So let me share my PowerPoint and get started. You can all see that?
Yes.
Okay. Hopefully this is going to work. All right. So as Wes said, I want to talk today about cell type expression phenotyping from deconvolution of bulk RNA-seq. And this group doesn't really need much introduction to the value of single-cell transcriptomics, so I really won't spend too much time on that. I just wanted to highlight the fact that one of the things that I'll talk quite a bit about today is single-cell data can help us find the frequency of cells in the population and look at allelic expression patterns for genetics, as well as finding markers of cell type. Monique is going to be talking later, the second speaker, that she'll talk primarily about single-cell EQTLs and molecular phenotypes. The human community is really going into this very dramatically, and she'll talk about a consortium there where they're looking at how you could move into personalized medicine. So I won't spend any more time on that. She'll talk about that quite a bit. I did want to mention a couple other things that really relates to the fact that we really are interested in trait-associated SNPs, but we really have to think about the biological context. And so, for example, there's disease-associated variants that often overlap with regulatory elements that are specific for a target cell or pathology. So if you think about SNPs that are associated with autoimmune diseases like Crohn's or multiple sclerosis, SNPs that are associated with those diseases are often found overlapping regulatory regions that are specific for cell type. So CD14 is cells that have expressing that protein are very important for immunity and autoimmunity, and also cells that like CD56, which is an NK cell marker or a T helper marker. Those are also quite important for things like multiple sclerosis. So the idea is that we really need to be looking at the cell level analysis here. Another example is if you have SNPs associated with red blood cell traits, they find that those SNPs are highly enriched specifically in erythroid cell regulatory elements. So we really have to be thinking about context-specific information. And people are already doing this in the human field, where they're looking at cell type specific EQTL in tissues and cell mixtures. So this is an example in PsycEncode, where they've been looking at single-cell genomics and regulatory networks. So they're collecting a lot of single-cell data, both at the level of gene expression for differentially expressed genes across cell types, but also regulatory elements and single-cell EQTLs. So they look at gene regulatory networks and trying to understand the regulatory networks that are controlling expression in specific cell types and come up with a linear network of cell type phenotypes that would allow them to predict phenotypes that could be associated with a multifactorial disease like brain diseases, and trying to predict phenotypes in the brain. The point I'm trying to make here is that this is a very large project with a very large amount of support. And so we can be thinking about this as an inspiration on the agricultural side. So what can we look at from the agricultural side? What we're interested in is we've received funding for comprehensive mining of the blood cell transcriptome for improved phenomics in swine. And I'll just take you through some of the reasons why we're working with blood. It's a very practical sample. It's a useful phenotype. It can collect it multiple times, and it doesn't have to euthanize the animal to do that. But it really needs improvement for use in predictive genetics. It's a mixture, a very complex mixture of cells, and there's a mixture of genetic effects, both cell composition effects as well as gene expression effects. So we're really interested in using whole blood RNA for another reason, and that's because we already have shown that if you look at whole blood RNA, it can be associated with disease phenotypes. Now, this is not letting me. I'm trying to get rid of this, but anyway, trying to move it. Maybe I can move it over. There we go. So what we've done is I want to introduce you to the Resilience Project. And so the idea behind the Resilience Project is one of the problems with breeding and trying to identify animals that are more disease resistant is the animals that we're using to create the next generation are all in a very clean environment for obvious reasons. We don't want to introduce disease into our nucleus herds. So we have to have the animals always in a clean environment, but we want to know what they're going to be like in a dirty environment. So this project is set up to test whether we can measure things in the clean environment that will be predictive of dirty environments. So we collect blood and a number of other phenotypes, including complete blood count, from animals that are coming from a number of different breeding populations, breeding companies. We keep them in a quarantine for a while, collect those blood samples, and then we move them to more of a normal commercial environment, which has a lot of natural disease challenges. And then lots of phenotypes are collected on these individuals, which are what we call the resilience phenotypes. So an animal that has ability to grow and do well in such an environment. And so we've collected over 2,400 samples in this approach. And so we have genotype for all these animals, as well as all these phenotypes. And initial work on whole blood has shown that we can look at quantitative analysis of the blood transcriptome, and we can see gene expression patterns that are associated with disease phenotypes. And this is a very big table. I don't expect you to follow it, but pulling up just some of the data from that, the traits that I'm showing here are things like health status or health measures, health scores. So these would be things like how many times an animal was treated and what was it treated with, as well as things that will be more familiar, things like growth rate. And you can see that there's a number of genes whose expression has been associated with these traits, anywhere from 100 to 400 to even almost 800 for growth rate. So that's very encouraging. And what's interesting is that we also collected CBC at the same time. So when we adjust all these samples for equivalent cell composition using CBC in the model, we can see even increased in number of genes that are associated with traits. We also see a decrease, especially for this health trait, and that indicates that some of these genes are probably associated with cell composition rather than gene expression. So this gives us some clues that this will be useful to us, but we want to go a little bit further and do a little bit better job in splitting up the data by cell type. So the basic general hypothesis is that tools for accurate deconvolution of whole data, whole blood data, into cell type specific transcriptomic data will substantially improve our molecular phenotypes. And we could then use them as direct selection tools or potentially as markers for animal traits. We have genetic variation, we have the collecting the blood data, and then hopefully having cell composition and collecting molecular phenotype, which we then associate with economical phenotypes. And essentially what we're starting with is thousands of whole blood RNA-seq data sets, which we then hope to convert to cell composition and cell type specific RNA expression phenotypes to get into cell type specific eQTL mapping. And this is stuff that Monique will talk quite a bit about. So I want to take a step back and introduce the topic of cell type deconvolution. And a great way to do that is with this review article that's really, can't recommend it enough. It was published last year. They did a number of important things that I think really make it great for resources for our community. Essentially, they were able to use a number of different deconvolution methods, created a method to compare all those using a very, very detailed reference data set. So I'll go through some of the background and talk about what they were able to see. So just in general, cell type deconvolution, what is it? It's a computational method that's been developed to infer cell type compositions and gene expression profiles within these heterogeneous or mixed samples. That allows us to better interpret disease mechanisms, immune response, transcriptional states, and cell proportions in disease phenotypes. And so the idea is that we have all these different biological samples, which are a mixture of cell types, and we're not following those if we look at bulk RNA-seq. So with deconvolution, we can get the cell type proportions and then cell type specific expression profiles. And there are two general methods, reference-free methods, which work in specific ways or specific situations, but I won't talk about that very much because reference-based methods actually are very useful for a lot of different approaches. And so the basic idea is if you have a reference based primarily from single cell data, we can take that and use it in bulk mixtures to deconvolute samples. So talking about a little bit more about the computation, the assumption here is that the total gene expression in the bulk sample is a linear combination of gene expression in individual cell types weighted by the cell type proportions in that sample. So the model is M equals S times F. And so what are those? So the M is the mixture. It's the number of genes in the sample times the number of samples you have. The signature matrix is the genes by the cell types. And the cell fraction is the cell types in the sample by the samples that you have. And so thinking about every gene in the bulk sample, you're trying to calculate a measure of the gene expression of that gene in that sample. So it's a product of the proportion of the cell type that's being measured by the gene expression in that cell type, and then you go down to the next cell type and get the expression of the gene across all the different cell types. The output will be cell type proportion because you can calculate that based on having a signature matrix and having the mixed sample, but you can also get gene expression profiles. And so the crux of this, of course, is if you have only the mixture, you really need a signature matrix that where rows are genes and columns are the cell types. So how do we create that cell type expression matrix? And I'll talk about that. Essentially, once you have a cell type expression matrix and you use it to deconvolute samples, there are several different methods to actually validate those results. And so one method is different starting data. If you have single cell RNA-seq data for relevant samples, you can create what's called a pseudo bulk sample by keeping track of the cell types in the sample. So you have ground truth proportions, but merging this data into creating a simulated bulk samples, which can then be deconvoluted with your cell matrix and then predict the cell type proportions, which can then be checked against the ground truth. Another method is to start with not single cell data, but cell data from independent cell lines that can be mixed at different ratios Then you have that mixed sample, which can be sequenced, and you have bulk data then. But because you kept track of the different ratios that you mixed, you have ground truth proportions, and you can compare that to the predicted that has been predicted from the deconvolution of the bulk sample. Another way that you can do this is, instead of having cell lines, you can take a sample that you want to deconvolute, so like our blood samples, and we have our bulk, but we can also separate the blood sample into the individual cell types. Those cell types can all be sequenced individually, and then you have the ground truth of the original blood sample because you measured the dissociated samples that you have, and have the ground truth proportions, but then also have the bulk samples, which can be predicted through deconvolution and then compared. And then finally, you can also start with a blood sample, separate it out into, using flow cytometry, into isolated cell types, each of which then can be measured for flow cytometry to give you the proportions. And you can also take those bulk samples, do the RNA-seq, and then use deconvolution to predict the cell type proportions, which can be then compared to the proportions that were predicted by flow cytometry. One problem with this method is that you're now using flow cytometry to predict proportions, and the gene expression used for deconvolution may not be perfectly collinear with the exact cell types that are predicted from flow. So that is one issue with that approach. So back to what they did in this review, they had a terrific data set that they can use to measure all these or test all these different deconvolution methods. There's 53 different methods that they tested. Their data set that they used to create their reference comes from Tabula sapiens and Cell by Gene, where they had a total of 63 donors with 30 different tissues and almost 300 different cell types. So what they are able to do is they have single cell data on all of these, using all the different tissue types from one donor, creating a reference data set and creating signature matrix of cell types by genes, and also identifying cell type specific markers so you can do yet another analysis for validation. Then they took all the rest of the donors. They created ground truth proportions by simulating different combinations, creating artificial bulk samples, and then predicted from using the signature matrix in the different methods of deconvolution, and then analyze the results compared to predicted and ground truth. And so then they were able to use one of their systems, this decon benchmark, to then predict how well each of these different methods worked. And that's really the heart of their contribution, is that they containerized every one of these methods, so it makes it much easier for anyone else to use those methods, and also an R-based decon benchmark that anyone can use. So they did this. They evaluated all of these different methods using this multi-tissue reference. And the other thing that I wanted to mention that I think is really fantastic that they did was they also provide a curation of a path to choose for your specific situation. So for example, we have bulk data, and we also have reference data for constituent cell types, as you'll see in a minute. We have single cell data and sorted cell populations, so we can create cell type expression markers. And so that tells us that we can use anything in this particular group of tests. And so we've decided to use CyberSort and AutoGeneS, partly because the running time is very short, very positive there, and it also doesn't take much memory, so it has high usability. All of this is contained at their GitHub, and so you can download that or open that up and download this information and do your own benchmarking of any of the samples with your own data. So I just wanted, in the minutes that I have left, just talk a little bit about where we're at with our whole blood RNA-seq data. So what the first thing to do is to create that cell type specific signature matrix. And so we wanted to create a new reference population or new set of reference data on immune stimulated pigs. What we did was collect PBMCs from a total of 15 different samples, as well as whole blood and transcriptome of 45 additional samples, overlapping samples, in fact, and then use single cell on these 15 samples, 5 pigs by 3 time points, and I'll show you that in a minute. We also collected from the whole blood data at the same time, collected flow cytometry analysis, so we can do that validation approach as well. So the single cell RNA-seq that we've done is on a Salmonella challenge, where we've collected single cell RNA-seq from blood from 3 different time points, 0, 2, and 8, as well as flow cytometry and quant-seq data. And the single cell RNA-seq data was collected using the PIP-seq approach, which was talked about in the first field day. And just to cut to the chase, we have about 100,000 cells that are passing the QC at this point, which we can then take into our gene signature matrix analysis. So what we've got additional data, as I mentioned already, we have single cell data from 7 samples and 13 different cell types predicted from that data. And then we have RNA-seq data from 9 cell sorted populations from 2 healthy pigs, covering all nucleated cells. And so they're shown here: B cells, T cells, NK cells, myeloid cells, and neutrophils. So we have all this data. We have 15 PBMC samples plus the 7 from prior work. We have the PMN transcriptomes. We have whole blood transcriptomes from quant-seq, as well as the 8 cell type flow cytometry from all those samples. So what we're doing now is doing the whole blood gene expression signature matrix using 2 different methods, AutoGeneS and CyberSort. And because we have collected all the matching data, we can then test whether our matrix is valid. I did want to emphasize this as a work in progress, and I'll show you only some of the initial work today. We have been using AutoGeneS, and I just wanted to go through a couple of reasons why we've been using AutoGeneS. It has a feature selection tool. It doesn't require predefined markers. And so the advantage of having an automatic feature selection is we can then use that method to create markers from the data itself. And one thing that you have to do is deal with multicollinearity. It's a critical problem in deconvolution when you're dealing with closely related cell types. It uses what's called a multi-objective optimization as a solution to select these non-collinear genes. The idea is that we don't want genes that have basically the same pattern because they will not be individually predictive of cell type. So what this MOO or multi-objective optimization approach does is that what we're looking at is trying to minimize correlation and maximize Euclidean distance. Because we have these different samples, the prior work, the sorted cell populations, and the flow cytometry, we can use a number of different validation methods. And just to give you a little bit of results on what we're doing and what we've accomplished so far is using AutoGeneS and the PBMC samples from the 7 sample. We've used the feature selection and identified a signature matrix of 400 genes. We wanted to check to see that that matrix was separating the cell types into data sets that you would expect. And so we wanted to see how correlated the gene expression is for different cell types. And it was gratifying to see that the cell types are correlated as you'd expect. So B cells are very highly correlated with antibody secreting cells, but not completely correlated, so we can separate them. And the same thing for the rest. So you have myeloid cells as well as T cells and other lymphocytes. You can also look at a heat map where you can identify genes that have high expression in white for particular cell types. And you can see that we have sets of genes that appear to be specific for each of the rows here. We do have one set of genes that is the best we can do for 2 different cell types that are very closely related. So we use that gene signature matrix from the 7 samples to then predict the proportions of the new 15 sample single cell RNA-seq data set from our Salmonella infection. And we pseudo bulk those 15 single cell data, and then we ask how well does the signature matrix predict the cell type composition of those single cell data sets? Because we know the truth, the actual proportions from the single cell data, and then we can look at predicted. So we can look within sample, but then across all cell types, and the Pearson correlation ranged from about 0.7 to nearly perfect for across the 15 samples, just showing you a couple of middle range samples, anywhere from 0.76 to 0.9 in terms of correlation. This looks pretty good, but it also doesn't really show you what's happening with a very low abundant cell types. And so you can also look across all the samples we have for each cell type, and gives you some idea of how the cell types are doing. So in some cell types, it's quite good correlation, 0.96, but it's over predicted compared to what the actual is. Some cell types are actually just quite good, and some are under predicted, but still the correlation is high. Some are sort of medium correlations, and then there are some that are very, very low abundance. So these are all less than 1% known, and it's very difficult to predict accurately those, and so their correlations are quite low. So just in summary, we have good predictions for most cell types. This matrix could be used to deconvolute the PBMC RNA-seq data sets for at least the major cell types, perhaps not the very low abundant ones, and so we need more work on those. For the whole blood, we need to add the major cell type that's not in PBMCs, the PMNs, and that's what we're really working on now. So we want to finally create a gene signature matrix using the PMNs and the PBMCs together. And we have two different truth data sets, the salmonella and the flu samples I mentioned before, but also an older data set where we have flow cytometry as well. We'll also test other tools such as CyberSort X. And again, we can use those purified cell populations either as a separate signature matrix creation or as verification. Then we'll go ahead and deconvolute our data sets, which we have a really large number of those quant-seq data sets. And we'd like to eventually develop a shorthand version so we don't have to go through all the steps in terms of RNA prep, quant-seq, and bioinformatics by using nanostring, which would allow us to do this kind of deconvolution work at much lower cost. So just finally, I'd thank all the people in the group that have been working on this project. Hoover and Ryan did some of the early work and collected some of the single cell as well as the sorted cell populations, along with Jane Wearda, who did a number of analyses of that, who was a PhD student in Crystal Loving's group, who's the immunologist on the project. And then Carrie Meeks collected the more recent single cell data on the Salmonella project. Mehek Kapoor helped analyze that data, and Muskan's been the leader on trying different bioinformatic approaches toward deconvolution. And this is a long-term collaboration with Jack and Graham Plastow on the Resilience Project, and Joan Lunny, who's helping us moving toward a cheaper and better approach to deconvolute whole blood data. So with that, thanks for your attention, and I think we have time for a few questions before we move on.
Any questions before we move on to the next speaker?
I don't see any in the Q&A.
I do not either. So perhaps we'll wait and get started with the next speaker, and we have plenty of time at the end, so please put your questions into the Q&A so that we can have both speakers address those questions at the end of the next lecture. All right, so the next lecture will be given by Dr. Van der Weest, I think it is. I'm not sure I'm getting the pronunciation correct. And she's going to talk about the single-cell eQTL consortium framework. It's a federated pipeline for genotype to phenotype-based association analysis. So some of the points that Chris made in his talk about a lot of these pipelines and how do we use them accurately and efficiently, Monique created some of these, and she's going to talk about that consortium's experiences. So Monique, the floor is yours. I see your slides are up.
Okay, cool. Thank you. Yeah, sorry for being a bit late. I didn't realize that it was actually 4 p.m. my time, but happy to be here today. So I would like to discuss with you our ongoing efforts in the single-cell eQTL Gen Consortium. So it's a consortium that we have set up to harmonize the pre-processing of single-cell EQTL data sets in such a way that we can do one big federated meta-analysis. And I would like to share with you the first results of our first data freeze, and also some of the more organizational aspects of the consortium that you guys maybe want to hear about as well. In our department, we're very much interested in genetic variation and how that may affect an individual's risk to develop a particular disease. So one way to identify these genetic variants is through so-called genome-wide association studies. And these GWAS studies have been very successful in identifying over 275,000 different genetic variants already that have been associated with developing 200 different types of diseases. The main issue with these GWAS variants is that it's not very clear how they are linked to the disease, because that's not really what a GWAS is telling you. So we actually believe it's very relevant to understand how genetic variants can lead to disease. So through which genes they may act, and in which particular pathway, and in which particular context. Because if you understand that, it becomes much easier to treat patients or even prevent disease in individuals at risk. So one way we can actually deduce what a genetic variant is doing is by just looking at where this variant is located. So for about 5 to 10% of the variants, we know that they are located in the coding part of the genome. And so it's relatively easy to then deduce which gene might be affected. But for the majority of the disease-associated variants, they are located in the non-coding part of the genome. So we need a different strategy to find out what they might be doing. And a very successful strategy that has been used by us and many others is something we call a quantitative trait locus analysis, or QTL analysis. So in a QTL analysis, you link individual genetic variants to a molecular data layer. And that can, for example, be DNA methylation levels or gene expression levels. And by then doing this type of QTL analysis in the different molecular data layers, you start to get a feeling of how a genetic variant through the different molecular layers may eventually contribute to a disease phenotype. So this is something that we have been doing quite a lot, especially focusing on expression QTL effects. And especially in the early days, we were mostly using bulk expression data for this. So we had initiated a very large international consortium called EQTL-Gen, in which we had combined the whole blood bulk expression data from about 32,000 blood samples. So this was both bulk RNA sequencing and microarray data sets together. And what we did in this data is that we did something we call cis-EQTL mapping. So defining whether a genetic variant is affecting the expression level of a nearby gene that is within a 1 MB window from this genetic variant. And so depending on the genotype of an individual for this particular genetic variant, the expression level of, in this case, the gene Y is being modulated. And doing this type of cis-EQTL analysis in this data set, we identified that almost every gene is somehow also genetically regulated. So for 17,000 genes, we identified at least one genetic variant that modulated the expression level. But what we also realized quite quickly is that having just these EQTL analysis conducted in the whole blood might not be ideal, because many of these EQTL effects are also very tissue specific, and going even one step further, they can also be cell type or even context dependent. And so realizing this, we also started exploring single cell expression data. So about 10 years ago, we started exploring, can we use single cell expression data also to do EQTL mapping analysis? The answer is yes, we can. And what we observed is that using the single cell expression data, we can actually find many EQTL effects that are cell type and sometimes even context dependent. So I give you here an example of a more recent study in which we observed this particular genetic variant to affect the expression level of SMID1. And since we did this EQTL analysis in PBMC data, we could easily just extract those cells that were monocytes. So we averaged the expression level of all cells that were a monocyte and observed that depending on the genotype of an individual, the expression level of SMID1 was either a bit higher or a bit lower. So we exposed these PBMCs to different pathogens. So we had an unstimulated condition, but we also had PBMCs exposed to 3 hours or 24 hours pathogen stimulation, and then having different pathogens. What we realized is that this EQTL effect for SMID1, it's present in unstimulated blood, and it remains active as an EQTL effect after 3 hours pathogen stimulation. But then after 24 hours pathogen stimulation, the monocytes didn't show any EQTL effect anymore. And since it was single cell data from the PBMCs, we could also easily check what would happen in another cell type. So when we did this also for CD4 T cells, so the same genetic variant, now taking the expression of CD4 T cells for SMID1, we saw that this EQTL effect was also present in the unstimulated T cells, but it remained active also after 24 hours pathogen stimulation. So clearly indicating that it's the cell type and the context of stimulation that can define whether a genetic variant is acting as an EQTL. So that made us realize that it's very important to actually use or take advantage of single cell data to define in which particular context and cell type a disease-associated variant might become relevant. So based on this, you might think single cell is the way to go. And while I believe that is the case eventually, we're now in this in-between stage where we have enormous amounts of bulk expression data and, well, single cell expression data is being generated and we get more and more, but really there is so much more bulk data out there, it would be a waste to not take advantage of that as well. So if we would compare equally sized single cell expression data with bulk expression data, and we would do EQTL mapping on that, then in this specific example here, we actually observed almost more than 40% additional EQTLs if we used the bulk expression data as opposed to the single cell expression data. And this was really matched samples. So clearly indicating that, yes, bulk expression data does have way more power than single cell. So that's why we also didn't want to stop with the bulk EQTL Gen consortium completely, because we still felt there was also a value for this bulk expression data. So we continued with a second phase of this EQTL Gen consortium that is now being led by my colleague Robert Warmerdam and one of our collaborators, Urma Vosa, at the University of Tartu. And they have now expanded the number of samples for which they have done EQTL mapping to over 43,000. And they have now developed approaches that allow us to also do trans-EQTL mapping at the genome-wide scale. So a trans-EQTL just represents a genetic variant that is affecting the expression level of a gene that is further apart or even on a different chromosome. And while doing these analysis, they realized that they could have identified trans-EQTL effects for almost 75% of the genes. But when doing this in the bulk data, they also again encountered a different challenge, being that it might be difficult to distinguish true regulatory effects from cell type composition effects if you're only using bulk expression data. And why that is, I would like to explain to you here. So when you identify a trans-EQTL effect, then this genetic variant is associated with the expression level of a gene that is somewhere else on the chromosome or further away. And what if this gene is, for example, a marker gene that is specific for a particular cell type? Then what could happen is that this variant is actually affecting the composition of the blood. And thereby what you measure in the bulk sample is a change in marker gene expression. But in reality, it was just a change in composition. And so in order to be able to distinguish between a change in composition and an actual change in gene expression levels, single cell data can again be very valuable. So now considering that individual single cell data sets don't have that much discovery power yet, we also felt the importance of actually having like an EQTL-Gen counterpart, but then for the single cell data. So that's when we started to initiate the single cell EQTL-Gen consortium, in which we developed pipelines that would allow us to, in the end, combine the EQTL summary statistics of many cohorts together. So in this first phase of our consortium, we have now 14 different cohorts, totaling over 2,000 donors. And we did this EQTL mapping in the 6 major PBMC cell types. And so for every cohort, we have phenotype data, genotype data, and single cell omics data available. And this is a small overview of the data sets that we have currently available. As you can see, for the majority of cohorts, we have about 100 donors. And per individual, we have around 500 to 5,000 cells on average. And looking at the technology, the majority of the data sets are generated using 10x genomics technology. But there is one data set with full-length SmartSync2 data, and we have one data set that has generated the data using the 5' end instead of the 3' end 10x technology. And it's also important to realize that even though it's all 10x technology, it can still be different version chemistry. So there's still some technical variation at that level. So this is the overview of the computational pipelines that were developed by the consortium. So we kind of organized it in such a way that different aspects of the pre-processing, quality control, cell type annotation, and EQTL mapping were organized in different working groups that were led by different people. So I'll go in a little bit more detail in the coming slides. But the idea was that we could pre-process all the data in a standardized and harmonized manner, such that in the end we could combine all data together to do EQTL summary statistics meta-analysis. So all these pipelines were built in SnakeMake, and the software is provided in Singularity images. So that allows that it's fairly easy for someone to actually run the pipelines. And we tested these pipelines in different high compute cluster environments, such that it was, well, most issues that could occur for someone were already solved beforehand. So in this first work group, we focus mainly on the pre-processing of the data and the quality control. So this working group is led by Martin Millet and Martin Hamburg. And yeah, it focuses both on the pre-processing and quality control of the genetic data and the single cell RNA sequencing data. So going into a little bit more detail here, the majority of the individuals in our cohorts were genotyped using genotyping arrays, so not using whole genome sequencing. And as such, imputation of genetic variants was also an important step. And for this, we used 1,000 genomes reference genome. And a specific Minimax imputation software was used for the imputation step. Then since it's good practice to actually multiplex multiple individuals in one sample pool when generating single-cell RNA sequencing data, This was also often the case in the cohorts with whom we worked. So one reason to do this is that it becomes more cost efficient to generate your data. You are better able to distinguish true biological effects from technical artifacts, and you also have the possibility to more easily identify and remove doublets from your data. So the consortium developed a specific pipeline for that, called Emaxify. And finally, in this part of the work group, we also calculated the quality control metrics and defined the threshold at which we want to throw out cells. So this was mainly based on the number of UMIs and the mitochondrial RNA percentage of a cell. So for us, it was also important that the individuals that were contributing to the individual working groups were also getting something out of it more directly, in the sense that they were also publishing parts of the pipeline in separate publications. So for example, what I show you over here is this Emaxify pipeline that kind of made a framework, like a decision tree. Okay, what should I do? How should I do the demultiplexing and the doublet identification for my data set, depending on the characteristics? And so as you can see, depending on the characteristics of the data set, it's a different set of different tools that were already developed that are well, working best in that situation. Then in this second work group, we were focusing on the cell type classification. So this effort was led by Joseph Paul and Ahmed Mahfouz. And the idea here was that we didn't want to use cluster-based cell type annotations, but we wanted to take advantage of these automated cell type annotation tools. Well, one reason for us was that if you want to make sure that the cell types from, if you want to make sure that the cells are annotated in a similar manner across all the different cohorts, automated cell type annotation is really beneficial. And these automated annotation tools, it doesn't matter how large your data set is, the accuracy of annotation will be the same. And so that was the main reason for us that this was the way to go. So how these automated tools work is that you have you require a very large reference data set in which the cells are already accurately annotated. And then you train your automated cell type classifier on this reference data set, and it can then be used to classify any new cell that is present in your own data. So while we were working on tools for this automated cell type annotation, we were working on a tool called hierarchical SC-Pret, but at the same time also the people from the Seurat package developed a tool called Azimuth. And so we decided, well, maybe it's a good idea to have the cells annotated using both tools, and then only select those highly confident annotations in which both tools actually agree with each other. So when we did that, we lost about 20% of the cells. So 20% of the cells was differently annotated with one tool or the other. And what is also important to realize here is that the annotations were quite accurate using unstimulated blood. And that's also in this first data freeze what we have meta-analyzed. But already looking at data sets in which we had, for example, pathogen-stimulated blood cells, we noticed that these automated cell type annotation tools are not perfect yet. So there's still room for improvement. So what we did here actually is that we annotated the cells at an intermediate level resolution. So for example, classical, non-classical monocytes, or naive or different memory CD4 T cells. And then we manually re-annotated it back to a low resolution, being, for example, a monocyte or a CD4 T cell. And only at this low resolution, we required both tools to agree with each other. Then finally, in this last working group, we were focusing on the EQTL mapping and the meta-analysis. So this is work that is led by myself and my colleague Mark-Jan Bonder. And what is important to mention here is that since we work with human data and human genetic data, it's often not allowed to share this type of information because it's privacy sensitive. And so for us, it was very important that we had a strategy that would allow us to let every cohort run all these pipelines just in-house, and that they would only need to share the summary statistics with us, such that we could meta-analyze everything in one centralized place. So the way we have done this EQTL mapping is based on analysis that Mark-Jan had done in the past, where he tried to find the optimal strategy for doing this type of EQTL mapping using single cell data. So he used a linear mixed effects model in which he uses he can add principal components as covariates and implements, for example, he can consider population structure and sampling variation as random effects in the model. Then he found out that taking the average expression level over all cells from one donor and one specific cell type works best, and that multiple testing correction using the story Q value was most optimal. So this was the type of things that we now have also implemented in our own single cell EQTL gem pipeline. And after doing this meta-analysis across these 14 cohorts, what we identified was about 6,600 e-genes for which we detect at least one genetic variant that is affecting the expression level. And you can clearly see that most effects were identified in the CD4 T cells and least in the dendritic cells. This is very much related to how many cells were available for the EQTL analysis, and also how many donors, because we had a few data sets that were sorted on, for example, only the CD4 T cells. So we really have also many more donors available for specifically the CD4 T cells. Then as a second step, what we could do is fine mapping to see whether there's maybe multiple genetic variants that independently can affect the expression level of a gene. And doing that, we found about 15,000 independent EQTL effects, so indicating that there's a little bit over 2 independent variants on average for every EQTL gene that we detected. Then looking at the cell type specificity of these EQTL effects, what you see here on the left is, well, in the middle, you see the amount of EQTL effects that were detected for a particular cell type. So for example, for the CD4 T cells, we detected almost 6,800 EQTL effects. And of these, a little bit over 5,000 effects could actually be tested in dendritic cells because there was sufficient expression level. And then going to the right, what we observe is that for those EQTL effects that could be tested in both the CD4 T cells and the dendritic cells, about we observed a correlation of effect sizes of about 0.59. So what you would expect is that cells from the same lineage, so for example, the lymphoid lineage, would resemble each other more than from a different lineage. And that's kind of what we see over here. So the CD4 T cells and CD8 T cells resemble each other most. And going to, for example, the myeloid cell types, we see that there's less correlation. Well, it's important to realize here that we did have the least power to detect EQTL effects in the dendritic cells, and that's why also there we see slightly lower correlations. Then comparing our EQTL results to the bulk EQTL Gen consortium, we were very much interested in that we actually also discover any new EQTL effects taking advantage of the single cell data. what we observed in blue is, so what we see over here is the total number of EQTL effects that were detected in single cell EQTL gen. And in blue, you see those effects that were replicated in the bulk EQTL gen consortium. And in orange, those that were not replicated. And as you can see, there is a fraction of the EQTL effects that could not be replicated. And when we then look at the cell type specificity, so whether this EQTL effect was only identified in, for example, the CD4 T cells, or whether it was more commonly identified, we noticed that especially those effects that were not replicating in the bulk data, they were much more cell type specific than those effects that did replicate. So it could also indicate that we're actually, that these additional EQTLs that we actually identify are true EQTL effects, but that they were just not discovered at the bulk level because maybe another variant was assigned as being the likely regulating variant just because it's a bulk composite of all these cell types together. Then to actually show that indeed there is a real value of these single cell EQTLs over what we have identified in the past using bulk EQTL gen. What I show you over here is the what is it? Enrichment of bulk EQTL effects for any GWAS trait. So we see about a 3.4-fold enrichment of EQTL effects derived from bulk for any trait. And specifically looking for immune traits, so immune diseases, for example, we see almost a 5.2-fold increase. So clearly indicating, okay, what we capture using the bulk EQTL gen EQTL results is somehow already relevant to understand disease, especially immune-mediated diseases. Now going to the single cell EQTL results and compare that to the bulk EQTLs, we see that there is an additional increase or an additional enrichment of overall GWAS traits, but also specifically immune traits by considering the EQTL effects that were only detected in single cell data and not in the bulk data. So clearly indicating that it's likely these cell type specific EQTL effects that are actually relevant for understanding disease. And so that's why there is a clear benefit of doing these type of EQTL analysis in single cell data. So what I have presented so far is this single cell EQTL gen pipeline, and mainly focusing on this first bit on the EQTL mapping and how we can then combine everything through meta-analysis. But we believe this pipeline is actually useful for a lot more than just EQTL mapping. So we made it in a way actually modulary, such that after running working group 1 and 2, we can, instead of doing EQTL mapping, we can also do, for example, cell type composition QTL mapping, something that we call co-expression QTL mapping, which allows us to identify the upstream regulator of an expression QTL effect, or do differential expression or differential variability analysis associated with particular donor phenotypes. So on all these aspects, we are now working to kind of expand the amount of interesting insights that we can gain from the efforts that are in the single cell EQTL gen consortium. And we also found it important that the frameworks that we have built are not just useful for blood PBMC data sets, but can also be used in other contexts. So this same framework of pipelines, we have now also made it such that it is like transferable to other tissues. So we have already applied it also to the brain in something that we call the single cell meta brain consortium. And We now also have both bulk expression and single cell expression data available for the blood and the brain, which also now allows us to start comparing EQTL effects across the different tissues and across, well, taking advantage of bulk data and single cell data at the same time. So with this, I would like to conclude my talk. So what I showed you is that these genotype to phenotype associations can be interpreted using QTL studies. And we believe that this is the right time to actually take advantage of both the resolution of the single cell data and this discovery power of the bulk data to get really the best of both worlds. And we believe that to do this in the best possible way, you need to have a lot of data available. And the only way we see this can be done is that the data is pre-processed and generate, well, ideally even generated in a similar manner, such that you can really combine all the data together in the best possible way. So what we would like to do in the nearby future within the single cell EQTL Gen consortium is that we also would like to expand our analysis to the higher resolution cell types. So far I only showed you analysis for the 6 major cell types, but where the real benefit will be over bulk QTLs for sure will be also in the high resolution cell types. So far in this first data freeze, we only focused on unstimulated blood cells, but it's actually there's so much more data available for both healthy and diseased individuals, unstimulated and pathogen stimulated cells. So it would be a waste if we cannot somehow combine this all together. And we're now exploring something that is called topic modeling to actually start integrating all this data together. So that takes advantage of how cells may have similar gene programs active, even though they are healthy or diseased and so on. Then we would like to also assess the impact of different donor phenotypes on EQTL effects, and through something that we call co-expression QTL analysis, and by integrating single cell multiomics data, we also would like to better understand the upstream regulators of these EQTL effects. So if you're interested in what we're doing, you can always go to this particular website. There's also a link, I think, to the GitHub, so you can have a look at the pipelines that we have developed. And with this, I would like to acknowledge the people that have worked on all this. So the people within our group, especially Roy Oele, Dan Kaptein, and Marina Gorshevniuk, have worked on the single cell EQTL data, and Robert Warmerdam on the bulk EQTL data. Then our collaborators in Tartu and the Munich Helmholtz Institute, and of course everyone within the single cell and bulk EQTL Gen consortium. So thank you for listening.
That's great. Thank you very much, Monique.
Welcome. Is there any questions?
So I have a couple in the Q&A, and there's also one in the chat just popped in. Actually, I think that all 3 are for you.
Okay. So the first question is that I mentioned at the end of my talk, cell type composition QTLs. So how many of the trans-EQTL effects detected in the EQTL Gen consortium are in fact due to cell type composition effects? So yes, we did do a comparison already. So the issue is, of course, that we don't have the discovery power as in the bulk consortium. Even though we have 2,000 donors in single cell EQTL Gen, we have over 40,000 in the bulk, so there's still quite a bit difference. So I think out of the what was it, 57,000 trans EQTL effects that were detected in bulk, only 1,000 could be significantly replicated in the single cell data. And then I would say around 85% was concordant. So indicating that, yes, many of the trans-EQTL effects seem to be true regulatory effects, but there is a fraction that we cannot replicate, so likely they are composition effects. So one thing that we already did in the bulk EQTL Gen consortium is correcting for some of the composition effects that were captured using principal component analysis. So that somehow alleviates a little bit this issue. Is that answering a little bit your question, Dominique? Well, I don't hear anything, so I assume yes. And then the next question, did you have among your samples for the single cell EQTL mapping also bulk RNA sequencing data? And did you check if using deconvolution, you could find similar results? So yes, we did have bulk RNA sequencing data available for some of our samples, but it was not collected at the same time. This specific comparison that I show you here, this was really like matched data, so really the same sample taken at the same time and then doing bulk RNA sequencing or single cell RNA sequencing and compare EQTLs. That was really same sample. For our own data, other people have tried to apply different deconvolution methodologies that have been published, taking advantage of the matched single cell data and bulk data, I believe. I'm not too familiar with the results, except that I know that the deconvolution actually works quite well for major cell classes. But as soon as you're going to look at cell types that resemble each other quite a bit, like CD4, CD8 T cells, and then specific subsets, it's getting more and more complicated. And also the more rare cell types, that's really where the difficulties are.
Yeah, maybe I could ask my questions, Monique.
Yeah.
Somewhat similar questions to Dominique. You talk about that bulk data is more informative, 41% more informative, but I'm wondering if that's because of the sequencing depth is probably deeper with the bulk, and it could be more informative in terms of the number of genes that are investigated. And then asking the same deconvolution question. I'm just wondering is if the bulk is really sensitive, and if you could deconvolute effectively, whether the best of both worlds would be to deconvolute the bulk. And then you'd have single cell level analysis that you could do.
Yeah, so going to your first remark or question, I think this 41% is just an example shown over here, and it very much depends also on how many cells did you use for generating the single cell RNA sequencing data. In this case, it's SmartSeq2 data. You tend to have, well, maybe a max of 100 cells per individual for SmartSeq2 data, but it is more deeply sequenced, and it's this full-length transcript data. It will be probably quite different for single cell RNA sequencing data that is generated using 10x genomics technology, focusing on the 3' end, where usually thousands of cells are available per donor. So yeah, I think the 41%, well, that can differ, but it's to just show that there is quite a big gap between what you can detect in single cell and bulk data. Then going to your last remark, I think it will always be like in an ideal situation, I think it will always be better to do single cell analysis, because then you really can also start taking advantage of um, the single cell aspect of the data. Right now, what I showed you is all averaging cells over donors and cell types, and then kind of pseudo bulking. But like the topic modeling approach or the dynamic EQTL mapping approaches that people are now exploring, they really take advantage of the heterogeneity in the data. And so it's something I think you cannot easily capture using deconvolution. And yeah, well, I think time will learn. But yeah, to me, the single cell data has the future, but yeah, and also, yeah, well, especially also, for example, in the multiomics data, what we see is that relationships that are observed between, for example, chromatin accessibility and expression level, they might be present when you average, but looking in individual cells, they might never actually, like observations might actually never co-occur. And this is something that you can actually figure out when looking really at single cell measurements, but having the bulk measurements, I don't think you can ever do that.
Okay.
So yeah.
My second question was kind of related to the, I think you maybe have addressed it, but I saw that you did have cohorts with very different number of cells per individual.
Mm-hmm.
Can you use that information to try to get an idea of what's the minimum number of cells that we need to collect on an individual to do such work?
Yeah. So our collaborators at the Helmholtz Institute, so Matthias Heinig, I think maybe 4 or 5 years ago, he did publish a methodology paper that I thought is also focusing on how many cells do I actually require to confidently be able to do differential expression analysis, but also EQTL mapping, I believe. Yeah, well, I can tell you that for dendritic cells, we may have about 10 to 20 cells on average for the data sets that have about 1,000 cells per individual. And I think that's really like the lowest you want to go. But for example, for other approaches like the co-expression QTL mapping that I have not really touched upon here, what we're doing is that we are calculating person-specific correlations between two genes over all the individual cells of a cell type and donor. And then you really use the single cell observations as a measurement. And then for us, it becomes much more important to have more cells per individual as opposed to taking a pseudo bulk averaging approach as you do for the EQTL mapping. So for co-EQTL mapping, 50 to 100 cells of a particular cell type are really needed to kind of get a confident estimate of these co-expression correlations at the donor specific level. So I would say it's also really dependent on what you want to do with the data. In general, people say for EQTL mapping, it's better to have more donors than more cells per donor if you can invest your money only once. But to me, the whole regulatory perspective that we can now also start to deduce from the data that is actually also a very interesting aspect that is more unique to the single cell data. And there, I believe we really need more cells per donor.
Thanks. That's helpful, as we think about developing projects in this area in the agricultural space.
Mm-hmm.
Yeah, it looks like there's two other questions, Monique, but you answered one of them already. How effective is deconvolution of bulk? And then the second one is, how do you handle privacy concerns? And I think you already mentioned that as well in your talk. Maybe you want to reiterate that part of it.
Yeah, so the genetic information we cannot share across different cohorts, or usually we cannot share that. So instead of asking the people to send over all their data to us, what we do is we distribute these pipelines that we have developed to each of the cohorts, and they run all these pre-processing steps and EQTL mapping on just their own data. And what they share with us is the EQTL summary statistics. So that doesn't contain any privacy sensitive data anymore. And through meta-analysis, we can then combine all the individual cohorts together. So it's really a meta-analysis of the EQTL summary statistics, and there's no need to share the genetic information.
So I have a question for both speakers in terms of the use of, obviously, we're learning so much from the immense discovery of all of these genes and these EQTLs that are cell type specific. Where do you see going forward the validation part of this in terms of using model organisms or validation in human using perhaps cell lines? Where do you see that going forward in terms of trying to really understand what we discover from these EQTL or gene efforts?
Well, I guess I could start. I mean, I think
Yeah,
the starting point for some of the validation of genetic effects is what you're mostly talking about. I
Yes.
think that at least in the pig and maybe some of the other animal species, is we look to see what's already in the human and mouse literature for genetic effects, and we usually try to see if we can see if there's been some similar association. There's been some good results where people are showing the same gene, or even sometimes the same portion of the protein is mutated in the same kinds of diseases. The humans and animals being so close. In terms of longer term specific validations, I think there are people trying to do such CRISPR type approaches in organoids and cell culture models of some of the organs we work on. But I think it'll be hard to do whole animal validation of very many of these effects.
Yeah, I agree with that. So something that we are also exploring is different experimental approaches, like these massive parallel reporter assays, where you assess for individual variants whether they have regulatory potential. So that then allows us to kind of break this LD structure that we have in the data and get closer to a causal variant. Then another thing that we take advantage of right now is the multiomics data. So having both chromatin accessibility and expression information from the same cell allows you to get closer to the likely regulatory mechanism that is underlying an EQTL effect. And I believe if you understand which, for example, transcription factor is binding to a location, and for example, the actual binding of the transcription factor is modulated by the genetic variant, it becomes also easier to pinpoint which variant is likely causal. Yeah, the CRISPR perturbation assays so far have, I think, only been conducted in cell lines. At least there's not much efforts going on beyond that. And usually it's still CRISPR-I, so that is like a more general transcriptional knockdown and not so much focused on the individual variants. So yeah, I don't think we're there yet, but these efforts combined will get us closer to the causal variant.
Great. I wanted to ask another question related to this accuracy of deconvolution. You mentioned frustration with rare cell types. You mentioned frustration with highly related cell types. As you know, the T cells share a common lineage, starting out with double negative, double positive, and then you go to all the specialized T cells, and then you get rare and rarer populations. And you mentioned this is problematic for, and DCs is one example you gave for this problematic association or finding EQTLs. Do you think it's going to be And first, I would say I agree with you. Everything, if we could do everything single cell, that would be wonderful because cost is so high that's not possible right now. But in the future, we hope that's the case. But in the interim, as we build out big and go back to legacy bulk RNA-seq data sets, how important do you think it is to try to, at least for future bulk RNA-seq experiments, develop the training set that's within the same sample repertoire that you're assaying for bulk RNA-seq?
So how much experience do you have with that, Christopher? Would you like to answer that one?
Yeah, I mean, I think one of the big questions, really for the longer term is how do we try to create the resources so that we can be more universal, and I think that's going to be a big question. Um, I I think the hard part about the future, to be honest, is how we'll be able to collect the data sets that we need to do that in the long run. So I don't know if you have something to add, Monique.
Yeah, so for us, we have not just been exploring cell type deconvolution using bulk RNA sequencing data, but also trying it in a bit more unbiased manner, trying to see whether there's particular principal components, for example, that capture variation in the bulk expression data that reflects cell type composition, but it could also be like the interferons activation within individuals and so on. So when you start correlating these principal components to QTL effects, then sometimes you start realizing that certain principal components reflect an aspect of the data that is correlating with genetics. And then you could start deducing by looking at which genes might be correlated with the principal component to actually see what might be the underlying process. So it could be cell type composition, but it could also be like a stimulation status or something else. And I think these unbiased approaches might also be relevant in a way because they can capture more heterogeneity in the bulk data than just the composition that you guys might be focusing on right now.
Yeah, our frustration has been with using some of the automated techniques. Of course, you can upload your own single cell data sets like cell typus, et cetera. And then there's data sets available on the human side, but they broadly categorize cell types and tend to cluster unique dynamic states of cell types into one. And it's a frustration because as we dig deeper into the details of trying to understand molecular mechanism, that state or subtypes of T cells, for example, is really important. And so we're trying to come up with better ways to clean training sets so that they're more specific for deconvolution of bulk RNA-seq. And we've, for example, looked at epithelial cells in the lung. There's like 4 or 5 populations. And if you go to human lung atlas, there's more. And so kind of where do you set those thresholds and how do you use model organisms to kind of validate some of that heterogeneity that you mentioned in cell type deconvolution?
So, well, one thing that we have done, so we call this methodology Piccolo, that can capture heterogeneity in the data that might be related to genetic variation. And well, the easiest principal components that we identify are, and that we can explain are associated with cell type composition. And a way to validate our findings is that we also have access to already the single cell expression data to kind of confirm that what we observe in the bulk expression data can be replicated in the single cell data. So for us, that is a way to validate these findings.
Okay, thank you.
Yeah, and Elizabeth asked me a question in the chat. Thanks for the nice talk to. Oops, it moved. To build the reference data set to allow deconvolution of whole population was quite an effort. How likely is it that potential future centralized resources will allow us to deconvolute bulk data sets of other independent studies, especially those with different breeds, pathogens, environmental conditions, et cetera? So that's with adequate resolution. So that's really a lot of what that Wes was asking about. I think maybe the quick answer to that is the human community, the human cell atlas community, and other communities are also dealing with that question, as Wes was saying. And the current concept is to try to create these foundational models that will try to give us the resolution that's relevant. It's really difficult to answer that question because it's an unknown and it could be different for every new data set. But the idea is, can you come up with a model that will allow you to map new data sets and identify where there's an unknown new data type, an unknown cell type, and then at least that would allow us to recognize that there's something new, and be able to try to explore that new cell type. At least that would help us. At least I think that's one of the main outcomes of what they're thinking about. But this idea of how do we define cells and how do we define cell type is an ongoing one. Much of our resolution is coming from gene expression, and so we don't have antibodies and other kind of protein markers that would really help us split those things apart at this point. In terms of just the reference, I think for a lot of what animal breeding is interested in and animal genetics, which I think is going to drive some of the funding of these things initially, I think we can at least focus on healthy samples as Monique's group has done as an initial starting point to show that we can develop the principles and the reference data sets that will be at least useful for predicting cell type and predicting genetic effects at the level of cell type in healthy animals or healthy plants. I think Sunita has put into the chat the survey that we'd like everyone to take. I think that there may be some issues, but you may have to reload the link. But the link works for me. I quickly ran and did the survey. It's really only 2 or 3 questions. So please take the survey, and we will also send this to the registered people that were not on this particular call. So you'll see that as well as the summary and the recordings available to you at a later date.
All right, I think we are at the end of our session here, and if there are no other questions, we want to thank both speakers, Chris and Monique. Thank you very much for sharing all your experiences with this problem we're all dealing with and trying to get to this better genotype to phenotype continuum using single cell type approaches.
Thanks, Wes. Thanks, everyone. Thanks, Monique.
Thank you.
Thank you. Goodbye.
Bye.
Bye-bye.